This is ENT student. The topic is Chapter 1 Molecular Biology Part 2. Cytogenetics and Molecular Cytology. Cytogenetics is a study of chromosomal abnormalities and the rearrangements. It usually has a major role to play in prenatal diagnosis such as Down syndrome and other congenital syndromes characterized by numerical chromosomal abnormalities. Theodore Boveri proposed that cancer arose from chromosomal alterations, proven with chromosome translocation 922, chronic myeloid leukemia, forming Philadelphia chromosome. Hybridization to target DNA in cells using fluorescence detection is known as fluorescence in situ hybridization. Interface fluorescence in situ hybridization has been applied to the solid tumor sections to assess the copy number of unknown sequence in breast, prostate, bladder, brain, lung, head and neck tumors. Fluorescence labels hybridization with cytogenetics is called as comparative genomic hybridization. Under red-green dual filter fluorescence microscopy and computer-aided image analysis, area of genetic neutrality appears yellow, underrepresentation appears green, that is tumor suppressed gene, overrepresentation appears red. Location of putative oncogenes. The picture is depicting the in situ hybridization. Polymerase chain reactions. Polymerase chain reaction achieves faithful and exponential amplification of a specific sequence of DNA by repeated cycles, each consisting of double-stranded DNA denaturation, hybridization of specific primers, and extension of polynucleotide by rapidly altering the reaction temperature between the segments of each cycle. The picture depicting the polymerase chain reaction. Step 1 denaturation, step 2 annealing, step 3 extension. These cycles repeat, resulting in exponential and accurate duplication of target DNA. A program of 28 cycles generates over 67 million copies. Polymerase chain reaction was originally described by Millis and Feluna in 1987 when practical problem prevented its instant exploitation. The DNA polymerase used in the original reaction was denatured during the DNA denaturation segment and therefore had to be added after each and every cycle. The solution came in 1989 when the lawyer isolated and characterized the DNA polymerase, Stag polymerase, from the thermophilic bacterium Thermus aquaticus, which normally resided in the temperature above 95 degrees Celsius. This polymerase was therefore heat resistant and did not need to be replenished between the cycles. Taq polymerase doesn't have proofreading properties. PFU polymerase incorporates a proofreading function to increase the amplification fidelity sequencing reactions. The genetic alterations in the tumor may not be present in the stromal tissue, and this total DNA extracted from the tumor will contain DNA from both benign and malignant tissue. This in situ contamination can now be eliminated by the use of laser capture microdissection. Loss of heterozygosity. Practical techniques based on the loss of heterozygosity of tumors DNA are based on Knudsen's two-hit hypothesis, which states that two genetic events are required to inactivate the gene that prevents the development of a condition. Inactivation of genes and oncogenic transformation. Inactivation of genes can occur by allelic deletion, genetic mutation, transcriptional silencing, conversion of proto-oncogenes to oncogene due to gene amplification. Conversely, a proto-oncogene may be converted into an oncogene by a simple increase in the copy number of gene, resulting in overproduction of protein by point mutations that affect the control of protein activity. Proteomics and Metabolomics. Proteomics is a profiling of proteins in cells and serum by two-dimensional gel analysis, separating proteins by charge and mass by X-ray crystallography and by mass spectrometry. Proteomics has the ability to identify post-translational modifications, some of which are cancer cell specific, which would not be detected by genetic or expression profiling. Metabolomics is a global analysis of metabolites but is a relatively new discipline. A mixture of techniques such as gas chromatography, high pressure liquid chromatography, capillary electrophoresis are used to separate metabolites, and the molecules are then identified using methods such as mass spectrometry. Metabolomics has already been applied to detection of oral cancer in saliva and serum. Telomerase. Critical telomerase shortening or inefficient telomere DNA repair can lead to both cell arrest and cell immortalization due to expression of telomerase, which, for example, has been demonstrated in 80% of oral cancers, 50% of leukoplakial lesions. Anti-telomerase drugs as a novel treatment remains promising. Mapping and identification of genes associated with diseases. The traditional approach to understanding the molecular biology of disease was to look for and characterize the abnormal protein. Diabetes mellitus and sickle cell anemia elucidated at molecular level in this way. Finding the protein when present in minute quantities is in an inaccessible site uh, such as the inner ear when nothing is known of its mode of action is however virtually impossible. Our approach to understanding such conditions relies on determining the chromosomal location, isolating and sequencing the gene so that the structure of protein can be determined. This is known as reverse genetic or positional cloning technique. The key advance that has made this approach increasing the successful has been the construction of high resolution linkage map of the entire human genome. Another potential source of candidate genes is to identify and characterize the genes that are expressed within a particular tissue. Genes expressed within cochlea, for example, represent the genes critical for cochlear function and thus are excellent candidates genes for hereditary hearing loss. Thank you.